I'm Dr. Maximiliano Figueroa. I am Associate Professor in the University of Concepcion in Chile, South America. And I applied to the fourth international call to visit, in this case, the CEITEC in Czech Republic to study interaction between molecules using the surface plasmon resonance technique. In our laboratory here in Concepcion, we are trying to develop the structural biology in different approaches, and one of our models is trying to study some molecules that are toxic, that are produced from some microalgae that produce the red dye that we call here in South America, and to check if these toxins can really interact with our engineered protein. We likely to develop the SPR technique here in Chile, so the surface plasmon resonance, that is not present in Chile in these days. And for that reason, we contact the people in the CEITEC because they have this molecular interaction platform that is very, very powerful, and they have their very nice SPR machines. And the idea was to try to immobilize our protein and then to test if the toxin can interact with our protein using that technique. And finally, we succeed. The idea was not only determine the interaction of the protein, but also establish collaboration with these people, because we would like to develop the SPR techniques here in Chile. We are now the only laboratory in Chile with the capacity to measure SPR. And finally, with the visit that we had in the past March and April there, I got the expertise to design experiment of SPR and immobilize protein on the different surface chips, and also analyze the data, everything related with SPR. So I started to check in the catalog of the Instruct-ERIC, and I found Joseph Hauser, and I saw that the facilities there are impressive indeed. I have to say, are really, really impressive, the facilities in CEITEC in Czech Republic. And I contacted directly to him and explained to him that there is this opportunity to me to apply to this call to get funding to learn and to measure SPR there and other techniques that are present in the CEITEC. So at the end was wonderful, everything. So we are very happy. Something very important for us was the opportunity to, through this project, have the opportunity to sign an MOU with the Instruct-ERIC. And this is something big, I will say, because here in Chile, we are just few laboratories trying to work in structural biology. So the infrastructure is, I will say, not so important, by say. The human resources are really, really good. All of us has formation in Europe or US, so we have this kind of formation and contacts. But however, we are lacking of the infrastructure. So through this project, we managed to design an MOU, and finally we signed this MOU between the Universidad de Concepcion and Instruct-ERIC, and we are now the first university in Chile we have signed this MOU with the Instruct-ERIC. So that is something very important for us as institution, but also very important as Chilean, because now we can have access to contact to the people there and collaborating together. So this is something, I will say, probably the most important thing that happened after this project that we realized together. I think this is for sure the outcome most important, the MOU that we signed.